Bonjour tout le monde. Merci beaucoup d'être venu en si grand nombre pour cette conférence spéciale aujourd'hui. So, with me wish to introduce our special speaker today, Tanya Berger-Wolf. So, I was contacted to see whether there was a possibility to make this conference today, because there's a special event, there's a meeting of this new center, which is the ABC, AI Biodiversity Climate Change Global Climate Center. That's held in Montreal, that was held in Montreal this weekend. And so they offered her to host Tanya Berger-Wolf to give us a presentation that is going to be named AI for Nature from Science to Impact. And so let me just introduce you quickly, Professor Berger-Wolf. So Professor Berger-Wolf did her PhD at the University of Illinois in Urbana-Champaign, and then followed with different postdocs, one postdoc in computational phylogenetic analysis at the University of New Mexico. Tanya was telling me that she started to work on the tree of life assembly just as she was doing this postdoc. And she switched to Princeton for a postdoc with Simone Levine. Yeah, that's it. And then she went as a faculty at the University of Illinois, Chicago. And then she is now the director of several institutes. She's director of the Translational Data Analytics Institute. She's the director of the Genomics Institute. I missed it.
So close, very, still get a sticker.
She's also the founder of the nonprofit organization WildMe. And in that case of today, she's the director of this new AI Biodiversity Climate Change Global Change Center. So I really have this picture.
You can also get.
So yeah, thank you for being here, Tanya. We're all very looking forward to see what you have to tell us. So.
Thank you for the introduction, and thank you for hosting me and taking time to come and listen about what's a good AI scientist like me doing in a place like this. There's actually, I'm not the only one, so I'm in good company of fellow AI and Biodiversity Change Global Climate Center, so AI and Biodiversity Change, ABC. Sorry, I'm trying to find the charter while we're at it. And so we're all, there's a group of us. I'll be main checking a few of them as I tell you about the types of things that we do and the types of things that that can happen at the intersection of AI and biodiversity science and why do it in the first place. So yes, most of the collections what? No, we're good. Most of the collections of these icons, I'll have them mentioned. So, and the ones that are not, we'll explain. So, I'm sure, especially in this audience, most of you have seen some version of this kind of report that we're losing biodiversity at an unprecedented rate. And that more than 1 million species are threatened with extinction. By the way, and this is a good audience to check, is 1 million a lot or a little? So how many species of plants, fungi, and animals, we're not counting bacteria, so no microbiome diversity. So plants, fungi, and animals above are out there. Lori, you're not allowed to answer the question. So throw out a number, like, you know, order of magnitude. What do you think? Any other? I heard a very specific 8 million. Are we okay with that number? Huh? 100 million.
We really haven't answered this question.
So they're good estimates, and yes, the uncertainty bounds are pretty big, but still they're about the order of magnitude of 10 million, right? That's a shockingly low number, actually. Like every time I'm like, yeah, really? I did not miss, I'm dyslexic. Did I miss a number? like, no, this is like the order, like, we now have several estimates, all come to around the same ballpark, at least the same order of magnitude. Okay, maybe we're off by like a factor, so maybe they're like 30 million. Still, this is a shockingly low number. 1 million is somewhere between maybe like 5 and 10% of all of biodiversity there, and it's threatened with extinction. And the thing is, of these 10 million, how many are named? Like down to species level? Anybody? Throw them up, huh? 2 million, the taxonomist in the room says. So yes, about 2 million are actually named. So we're losing species probably faster than we can even name them. In fact, this is the map of biodiversity loss. And it's also not only that we're losing biodiversity worldwide, the highest rate of loss is in the places where the biodiversity is highest. Right? This is the percent of biodiversity loss at this point. And so, yes, we know, and probably this room particularly knows that there are these five drivers of biodiversity loss. They're not independent, they're highly complex and interconnected in all kinds of complex ways among them, right? And it matters, the fact that we're losing biodiversity. I probably don't have to convince you, so no quiz time, like what do you think it matters and why do you think it matters for this audience? But the thing is that with all of that urgency, the fact that we're in the middle of the sixth mass extinction, if you look, our understanding, the tracking, the monitoring of biodiversity is really not that great. The International Union for Conservation of Nature Red List, so IUCN Red List, which is the official international organization that's tasked with monitoring the biodiversity of the world. They're literally named the Biodiversity Monitor. When we say that the species are endangered, it's because IUCN Red List Commission for that species determined, according to some metrics, that the appropriate status of this species is in the endangered bucket, and that comes with certain resource allocation and policy protection and other things. So remember, 10 million-ish or so? They monitor about 160,000 species currently, and of those 160,000, notice the huge taxonomic bias here, right? They're monitoring, their goal is to monitor, this is a little bit out of date, but their goal is to assess about 8,000, let's be generous, species of fungi. I heard there's a couple of people who are interested, who are like specializing in fungi here. How do you feel about that? Right. All right. We're clearly taxonomically biased here, but even of those 160,000 taxonomically biased species, more than 21,000, their conservation status is data deficient, as in we have nothing, not enough data to make even the basic determination about anything for this species. About 66,000 more, the population trend is unknown. So yes, we know we've assigned to some conservation status, but the population trend, up, down, stable, who knows? And these are not obscure species. Killer whales, the biggest dolphin in the ocean, hard to miss, data deficient, right? Polar bears, the iconic conservation species. Population trend is unknown. We think it's vulnerable as a status, ask an 80-year-old, they probably will tell you the same thing. So, but not to be too facetious here, the thing is that there was a crisis of data and ways of translating some data that we have already into some decision markers, decision ready metrics, right? And what can we do about it, really?
Well, actually, we can do something. This is where technology comes in, because over the last 20 years, there was an explosion in the planet's life monitoring technology. Right? We have remote sensing, which is pretty mature technology by now. We have autonomous vehicles underwater, on the ground, and in the air. We have on-body sensors that you can put everything from heartbeat monitoring and GPS to like monitometer to track the position, the location, sort of where the animal is looking. We have stationary sensors such as acoustic sensors and camera traps. And oh, this is thankfully, I think the audience, I don't have to explain what a camera trap is. Okay, good. And then we also have these guys. These are sensors, and these are really, really good sensors for nature monitoring. How many of you have taken a picture of a living organism in the last week? Today, how many of you have taken a picture of a living organism today? For those of you who have not raised their hand, may I remind you that humans are living organisms? Right. And even when we don't think that we're taking a picture of a living organism, we quite often do. There might be a picture of a tree, a plant, trees are plants, a plant, an insect, a spider, right, that we didn't quite notice when we took a picture, or a wing of a bird in the window or something. And then more recently, as a sensor, there is also environmental DNA, which is increasingly used to monitor biodiversity. But if we look at the data that we're getting from all of these sensors, the situation is not that rosy, right? So notice again, this is the map of biodiversity richness, essentially, the species richness, the biodiversity hotspots. And this is the GBIF map, the Global Biodiversity Information Facility. So there's not quite disjoint, but really there's a huge bias for North America and Western Europe, which is not the hotspot for biodiversity. And if you zoom in fact, and this is the same situation is, if you look at INAT, which is today the largest source of biodiversity, is termed as the largest source of biodiversity data, but it's hugely, hugely biased in many different ways, including spatially. And this is birds, eBirds data. So not only that it's biased, almost complementary to where the biodiversity is the highest. If you zoom in, it's it's biased to roads and urban areas. It's biased to where humans are, and not any kind of humans, right? Clearly, there are humans in Brazil. It's biased to in the US, for example, iNatural's data is biased to the income. Like you can correlate it with the income data. So not only we have a lot of data about very few species collected by very few very privileged people, but there are two takeaways from this. Everything we learn from this data, we keep on learning about the things that we already know a lot, and probably not the things that we need to be learning. But the other thing also is there is data there, right? Clearly, Brazil is collecting data on biodiversity of Brazil, right? But it's not being kind of available to the global biodiversity tool building, to the global biodiversity decision making, to the global biodiversity monitoring efforts. Which means the decisions that are made for Brazil are not the right, if they're made sort of at a global scale, or if the tools that are built do not are not the best decisions. All right, and what are the tools that are being built? So what happened also in the last 10 years about, and certainly in the last 3 too, is that there is an explosion of AI and machine learning specifically tools that can make sense of a lot of this data. So for computer vision, for example, we really can take an image and, you know, find all the animals in it, which is section, we can put a bounding box around the animal, we can, do species classification, as in what kind of species it is. We can do the kind of stick figure, fit a stick figure into it for pose estimation, so we can start doing behavior. We can even identify individual animals here and do environmental reconstruction. Many computer scientists will claim you that these tools are done, exist, here it is, use my tool. The thing is also, if we go back to the same biodiversity richness map, the hotspots of biodiversity map, and overlay the map of AI production as measured by publications and startups in the world. Yeah, again, most of the AI is produced in absolute absence of any kind of biodiversity questions context. It is completely disjoint and unaware. And more so, the culture of machine learning, of modern machine learning today, is completely wrong for producing tools that need to be used in a field, any applied situation, but particularly when they have to help, you know, address, understand, and mitigate an urgent and huge challenge with massive amounts of data that are completely not used, biased data that haven't been used in any way in the construction of these tools. So because, and this is the thing, that machine learning culture today is that we use this big stereotype benchmarks, and this is the work that's been led by David Rolnick right over there, who is here from McGill, from Montreal, and part of many of the things that I will be talking about. That, you know, not only is this big benchmarks and the entire value of your tool that you're building is how well it performs on that benchmarks. And we measure how well it with a very, very, very narrow set of evaluation metrics. And then you build a tool, it has to be a massive data set. And then everybody else evaluates their tools on the same massive data sets, and it has to generalize to everything else. Not only you should be able to recognize, to correctly identify novel species that nobody else has yet identified, but you should also, the same approach should be able to recognize not a new type of a water bottle and a car model or whatever else, right? So it should be exactly the same algorithm. So it's wrong, and this is what kind of the community, we need to build very different kinds of tools. So here's a couple of examples. And by the way, not only that we need to build, including, so there is now a growing awareness that the approach that machine learning has taken until now, including with the GPT and others and other modern type of machine learning approaches, that are now complaining they're running out of data. Like, well, let me show you a few million camera trap images. Come on. That not only that is inappropriate for the kinds of problems that come up in biodiversity, but in broader set of problems, including health and climate. And so there is a global partnership on AI. It's an intergovernmental partnership that is now became part of a think tank, OECD, which is the Organization for Economic Cooperation and Development, the AI arm of OECD, to really advise governments on AI from science by scientists, by the AI experts in different domains. And so including reports like this on AI and biodiversity that addresses this aspects of data, the challenges and opportunities that we have in data, like getting more data, getting data that's already there, filling these gaps in data, and then driving to understanding of the loss of biodiversity, the drivers, this complex interconnection of already complex drivers, and then putting in resources and bringing all the way to policy and action. And so the 5 outcomes that the recommendations that are not particularly surprising, but that we really need a global cooperation on biodiversity data to really bring together the existing sources. We really need to engage communities, local communities at every level, to be part of the process from data to decision-making, but particularly as we develop AI tools, we cannot just take data, develop tools, give the black box, and we do need to develop, we need resources. AI is expensive in many ways. Not only that the skill set is expensive, right? But it also, the hardware is expensive. The running sort of the initial purchase, running the algorithms, a lot of parts of it are very expensive. So it requires different type of resourcing. And that's where, yeah, finally we can get to the point of developing tools and metrics and then capacity building. So that the tools that we develop, I actually used, right? And not only used, but useful and used. So with that, this is where we think the AI and biodiversity change, so ABC, Global Climate Center, comes to kind of be part of addressing this challenge. And this is a research center co-funded by the US National Science Foundation and the Canada's Natural Science and Engineering Research Council. Yes! I get a sticker. And the goal of the center is to contribute AI-enabled tools and analysis that describe the impacts of climate change on species and habitats, and which can assess how they respond to policy and management intervention. And the whole approach, particularly in biology, is, so in biology, the questions that we pose, and so we have still here. So we had a meeting, all hands meeting, our first annual all hands meeting of the center. It's very new. We still have some people who haven't left. So from the co-PIs, so David Rolnick, who is leading the Canadian side, Laura Pollack, also from McGill. Is that it from the guys? Yes? Yeah, okay. Just the locals have left, like everybody. Like, they're like, okay, we know what you're gonna be talking about, that's fine. Anyway, so on the biology side, really the focus is on the change. The change and the hitherto unobserved, unknown, undersampled, like how do we get to the understanding of the species that were undersampled, that are cryptic, that are at the edges of their ecosystem, at the edges of the reachability of humans, the changing ecosystems, the ones that are most sort of maybe vulnerable to the climate change pressures and impact. And so there are several questions around it about the range shifts and movement with the climate change, the sort of understanding recovery of ecosystem as after catastrophic events, and understanding vulnerable ecosystems. And so on the computer science side, on the machine learning side, the set of tools like to address really this change, we really focus on how do we develop tools not for the bulk of the distribution. All distributions in biology, I found a long time ago, are these long tail things. We know a lot about very little, and then goes the rest of the data. We have a lot of things that we know very little about. And so how do we develop tools for that, for the long tail? how do we develop tools for the bias data? How do we develop tools that take advantage of all of these sensors and our ways of looking at nature that I showed at the beginning, and be able to put them together. And so, and with that, so here's a few examples. This may look familiar to some of you. It's in your backyard, literally, right here. So this is an automated monitoring of insects, so this is an insect camera trap developed by David's group here at McGill with in collaboration with Mila and other partners. And so not only it's the hardware that takes a picture of insects that are attracted to this white space, but then what do you do with this picture? And this is where machine learning comes in. Right, so insects, as you saw in IUCN Red List, and you can look at any other source of biodiversity data, hugely undersampled. We really, what is the quote that I don't know if God exists, but I know that he had an inordinate fondness for beetles. Not only beetles, but like all the other insects as well. There's a lot more of them, and we know very, very little. And so, how do they take a picture like this and not only name the species that are there? So you have to find each animal, you have to find the outline, and then figure out what species they are, hopefully. But also, there are going to be lots of new species, and one of the really, really cool things that are being developed is flagging potentially new species that machine learning methods are actually not good at, and that requires really, really deep innovation in machine learning to go from the 2 million named species to maybe 10 fast enough so we actually can know what's out there before we lose it. Right, so that is really focusing on the tool development aspect of this AI and biodiversity challenge. The other approach that David's group takes for filling this biodiversity gap, extending a gap in data, so the data part of things, is taking advantage of multimodality. Can we use one type of data to maybe predict another, right? Can we use the satellite data, satellite imagery to predict bird species? Can we predict butterfly observations from satellite data and bird species? Right. Again, sort of and then use butterflies to improve bird predictions and vice versa. And can we then improve maybe satellite data labeling itself, right? So really, because all of these modalities are really biased in very different ways. So if we can take advantage of the good chunks of one data stream to improve the other, to find these hidden relationships, let's do it. So that's one way of addressing the data gap. Another one, and this is also from one of the, from Sarah Beery, who is part of the ABC, is, so iNat, how many of you have used iNaturalist or at least heard about it? Okay, so if you haven't heard about that, that's the largest source of citizen science data. It's an app, you can use it to do observations of nature. It has, I think at this point, something like close to 40 million observations reported, hugely biased, but still, this is the best we have. And in terms of like good coverage, good spatial coverage. And so the thing is, people report an observation, and we typically focus, and those who report, when they report it, what they have in mind is whatever ground of the picture. But quite often, in addition to that, there's all kinds of things that are, information that is really already present in that picture. Not only what's the species and where it is, the who, where, when kind of thing, but what is it doing? Maybe it has another species in its mouth. A plant or another animal. Maybe there's intra-species interactions, like they're mating or fighting or just displacement or something. Maybe it's interaction with the environment. Maybe we're starting to see species where there shouldn't be. Maybe it's interaction with, maybe there's something in the background that we didn't even notice until now. And so how do we get to all of these questions that we didn't even think this data could give us answers for? And so this is a tool, Inquire. This is more of a survey of what kind of things, what kind of questions might you be able to ask of iNaturalist data that Sarah Beery and her group has built for iNet, and you can interact with it on your own and sort of suggest other questions that you might want to investigate, and then the system behind it will figure out, can you actually answer it using iNaturalist data? And so this is starting to build tools and engaging the community right through this sort of interacting. Let's source the questions that you might want to know the answers. And then going even deeper, sort of getting to really understand. So I've talked to many scientists, and I would love to do the same survey here, particularly when I talk to biodiversity scientists. So if I said, what's the one thing that you would want to know about species out there? One of the most common answers, and maybe I should, how about you? If I give you one thing, just one kind of however big, small, or whatever answer, but it's going to be just one question that you get to get the answer for. What would you want to know about species?
Its name.
Its name?
Yeah.
Okay, that's easy. We'll get there. We'll get there in the next slide. Yes. What else? Anything? Can you eat it? Can you eat it? I must be in a French adjacent place. Yes. Yes.
It's carbon content, right?
What is what?
The average dry weight.
The average
Dry weight, but the organism, like the carbon content.
Oh, yes, dry weight, the carbon content. Interesting. So far, nobody has asked the question that I get most commonly, which is the intraspecies variability, diversity, right? For any set of traits, what is the distribution within the species for that trait? So the intraspecies diversity. Would you want to know that? If I could give you the answer, maybe, would it be useful? No? Okay, well, I see some nods. All right, I'm not that far. Great. So the basis of a lot of these questions, even to understand, dry weight, or why the species doing something or not doing something, why they're here and not there, what's going to happen if, the basis of a lot of this is traits. Right? And so not only we want to be able to get at least images are by far the most abundant source of information from all of these sensors that I named before. And we're hugely visual species. So if we can extract something from already existing collection of images, if we could extract traits, we could start answering many of the other questions. And so that's the premise of Imageomics. It's another National Science Foundation funded institute in US, and it's a new field of science. And so so it's the omics, as in biology, the holistic aspect, right? The general biological information from images. So we got to practice. So it's imageomics. So say it with me. Imageomics. Again, imageomics. If you say it correctly, you get a sticker. Otherwise, you don't. So a new field of science, a new interdisciplinary scientific field focused on understanding biology of organisms, particularly traits and phenotype from images. And so really making biological traits computable from images and being able to not just look at this collection of images and being able to say, this is this species of bird, but this is this species of bird because, well, because it has yellow belly, black behind the beak, and it wobbles when it walks, and it makes this kind of call. Right? So wouldn't it be great? And then we could also, what if not only that, but we could also measure across all of the birds of that species, like how that varies. And so that's what we set out to do. Quick reminder for those who forgot what traits are, starting with the Mendelian P experiments. So traits are any morphology, color, size, shape, but also it can be behavior, such as playing for the possum, playing dead when threatened, or how a bird weaves the nest, or the patterns of zebra stripes. But it also it can be an individual. It can traits may change with disease or age, and demographic status, right? And it's a product of genes and environment and the interactions between those. So why do we think that we can do traits from images? Well, because we have massive amounts of images now in variety of spectra, and we have a lot of additional biological information such as text, molecular geography, other modalities, that modality part, and we also have a lot of the biologically structured knowledge that we can add, such as even the basic things like taxonomy and phylogeny, but also ontologies and other biologies, the science of structures, right? And kind of classifications. And so because of that, with particular framework in machine learning, knowledge-guided machine learning, we're hoping to come up with these answers of this is the species because. And we've started to do it. So we built the first foundation model for the tree of life. So this is a massive machine learning model that's trained on massive amounts of data without a particular, so it's a foundation for many other tasks. So it's not the task that it's, you train it one for a specific task, and then when you need to change it or adjust the task, you retrain the whole thing. No, the goal for foundation model is to serve as a foundation for many downstream tasks. And so this one was trained for the kind of baseline task of species classification, and trained right now is the version 0.5 on a million images for about 450,000 species. So getting to a quarter of all named ones. And you can even do it on your, try a demo with many fewer species, but still a demo on your phone, and you can take a picture and see if it will recognize it. Because it's taxonomy aware, taxonomy is built into the training, you can classify to not only species, but any level up. So from there, you can do zero-shot things, meaning that of pictures of things that it has never seen before. You can give it some options. And so, for example, beetle people, we had this event called Beetle Palooza. We call Beetle Palooza, where beetle scientists and machine learning people can think together and start building tools for beetle, all kinds of beetle tasks. And so building a tool that can identify identify beetles from picture to a tribe, and then sort of that pretty reliably, and that sort of is then from that point on, it allows a human to sift through many fewer options and to recognize, or maybe recognize that it's a new one. Again, so filling this biodiversity data gap. We also developed interpretable, so starting to add this knowledge to add interpretability in a form of, this is the painted bunting here, the important parts of the big image. So again, it really separates it into the head, the belly, and so on. So starting to build the tools. And so and pushing it even further, And this is tools that not only identify, given these collections of images, identify them to the species level, but really can go down to an individual, not just zebra, giraffe, whale, or turtle, but Zippy the zebra, Joe the giraffe, Terry the turtle, and Willie the whale. And this is for anything striped, spotted, wrinkled, or notched, even using the shape of a whale's fluke or the dorsal fin of a dolphin. So marine and terrestrial. And then with information on when and where the image was taken, you really can now use images to count and track animals. We built a platform, so realizing that it is useful beyond kind of our own curiosity of being able to identify zebras. We built a platform, started a nonprofit, WildMe, and built a platform called Wildbook. This is an example of a wild book for cetaceans, so fluke book. It's an instance of a wild book. And this is a page for Pinchy, the most cited animal in that wild book. Pinchy is a sperm whale, if you don't know your whales. She has been cited more than 600 times, hangs out around Dominica. I would too, if I could. And the thing is also you can really, by co-citing, you can start looking at the social network movement and many other things. So this is really, now you have tools and decision-making support with this kind of approach. So Flukebook today has actually close to 3 million images. The entire North American whale catalog from NOAA and BOEM and many other kind of sources is now on that platform, including, yes, Canadian government data. There are more than 80,000 uniquely identified individual animals in this. And about a year ago, so actually a little bit more than a year ago, we had a new species added. There's now, I think, 8 or 9, actually more, of whales, and then some more of dolphins. But the new species, any guesses what was the new species? This is the quiz time if you paid attention in the beginning. Thank you. Killer whales, yes. So killer whales were added to Flukebook just about over a year ago, and we're getting to the point where there is actually enough data now for the Species Commission to do reassessment and to change the conservation status from data deficient to something, right, for the first time. Like this is, and the reason is, we didn't collect this data. We don't own a single piece of this data, but we created technology that actually connects people, right? We actually went a step further. We built a social media intelligent agent, a bot, a very, very simple one. We're not using ChatGPT. We're not using all of the resources that come with the use of generative AI. No need. It's a very simple bot. It scans, finds pictures of relevant species, in this case, a whale shark, publicly posted on social media, on the particular platform here, and then sends it to, then actually posts in the comments saying, hey, at 2 minutes 46 seconds, we found this picture of a whale shark, MXA 700, here's everything we know about it. And essentially, and then engages the users and asks for permission so that they allow to post, and then also to add to the Wildbook, and then points them to Wildbook. Like, here's everything we know about it. And people engage. Most common response that we get is, wow, this is amazing. How can I help? Right? Because suddenly they connected to this animal, MXA 700, that they saw on their Cancun vacation that they had no idea could be useful for science or conservation, but just by making this connection, that this could be, like, here it is, here's what we know, here's everybody else who saw that animal, right? And at the bottom of this website, this is Sharkbook AI. This is Sharkbook. And the bottom we post everybody else who contributed to this data to this specific individual, because there is not one individual organization, not one research project that has the entirety of the information, even about one whale shark. These are global species, they travel 5,000 miles. right? It's only by putting all our data together, however small and local, however big and global organization you are, that we can truly build the picture of a global species. And we need to build AI that connects people, connects data points, connects people to nature, and engages the communities by design. And so Sharkbook today has more than 22,000 uniquely identified individuals. And the IUCN Red List entry for whale sharks was changed already in 2016 based on the data from Wildbook. And the most comprehensive study on the biology of whale sharks was published using data from Wildbook, co-authored by 37 authors who met through the pages of Wildbook. At the bottom of this, here's who contributed, that's how they met. And they realized that by sharing data, by bringing it together, they can understand the seasonality of migration patterns, because they didn't realize that this whale shark is the same in Cancun, is the same one that traveled 5,000 miles to Philippines, right? And so this shows the strength. Oh, and also based on that data, the conservation status of whale sharks was changed from vulnerable to endangered, and the population trend from stable to decreasing, not because the species are doing worse, but because we actually have the right data to make the decisions, to make that evaluation, right? Data, improve the assessment, improve the indicators, and that changes policy, changes the resource allocation, it changes the decisions about that species, right? All the way to policy. We have now data for more than, well, actually way more than 100 species. It's a hard stop at 5, right? I just want to not go over, right? Yeah, it's a soft spot. Stuff. Okay, so the same technology also helped count zebras in Kenya, the endangered gravy zebra. There's about 3,000 left. And for the first time in 2016, hundreds of people were driving around the country. 95% of the species are in Kenya. So driving around the country, taking pictures of every gravy zebra they saw. And 2 days later, 40,000 images. We did identified every individual that was identifiable, and came up with an estimate, population estimate. But the important part was that everybody participated. These hundreds of, well, about 200 people actually, the first event. It was everybody from school kids and local rangers to US ambassador to Kenya at the time, and tourists with telephoto cameras. We're taking this a variety of images, right? It was the tagline for the event of Kenya's powering conservation. This was their data, their decisions, right? They could trust the process. And Kenya Wildlife Service went complete 180 from saying this is not how we're counting zebras to, yeah, this is how we're going to count zebras from now on. You're on the hook every 2 years. Let's go, right? Wait, we're not prepared for that. But we repeated it in 2018, 2020, pre-pandemic, and then 2024, this past January. And so because the uncertainty bounds around the estimate were so tight, because every zebra essentially was seen. There was enough people to photograph every zebra that was out there. By the mid-morning of the second day, we were nobody was taking a picture of any new animals anymore, essentially. So the recapture rate, right, was super good. And therefore, the uncertainty bound, we had for the first time, it was the 2352 plus minus 72. And for the 2018 estimate, it was 2,800 plus minus 150. Meaning that we could definitely say, yeah, the population is definitely growing. The confidence bounds were completely disjoint. They could make decisions. They even made some really controversial conservation decisions based on those data and showing that it actually improved. And it was possible not because we had amazing algorithms, but because we engaged the community. We had cameras, GPS-enabled cameras, about 200 of them that we could give out. We came up with a protocol, very old school paper-based protocol to synchronize every camera that did not have GPS to the one that had GPS, right? We worked with local nonprofits that created a booklet that explained what was going on, and created whole competitions and the whole process. It like really explained the whole process, and went around the country to different communities to really do the training and explain. So this is the training and explanation and engaging the community in English and Kiswahili and in Samburu, northern Maasai, to make sure that everybody could participate. We also gave training to conservation organizations, conservation staff, so they could use Wildbook. And I like still don't have that t-shirt. I want it so badly. And that helped. It changed policy. The Kenya Wildlife Service species management plan actually was based on this event and was co-signed on these numbers, not on the event, on the numbers co-signed by all the governors that had Greve zebra in their counties. And Simon Kitao, Assistant Director for Kenya Wildlife Service, said, this shows the power of citizen science and machine learning for conservation. I nearly cried because that was the first time that Kenya Wildlife Service used the words machine learning ever. I verified. So that also changed the entry for IUCN Red List. So that also went all the way to policy impact. But the important part, it was possible because the community was engaged, not because the fancy algorithm. And so here, one of your own Montreal's own Andy Gonzalez and Laura Pollack really think that this pipeline from data to tools to inform decision and policy that then go back into informing sort of what other data and tools we need requires a global cooperation, bringing together not just, oops, we're having that we cannot, this situation where we have lots of data for a few things, and we keep on learning things that we already know in the first place because we've learned them already from that data. That's not the right way to go. So how do we bring that cooperation? How do we bring the sort of different data sources into the same contexts? How do we enable those who have their own local source of data and maybe do not want to quite disclose it, but give them tools that have been maybe developed on this other data and can be fine-tuned, like tweaked from foundation models, from this framework of foundation models, from multimodal approaches to help make decisions locally that then propagate to global decision-making. And this is the framework that they propose. And so hopefully, I gave you just a taste of how machine learning can be not only just an approach to speed things up for data collection, but also a tool for engaging communities to help make decisions that then impact us all. I clearly am not, I'm standing in front of you, but I'm not the one who did most of this. I speak for a community, represent for a large community of lab and my students in the genomics, and Biodiversity Change Global Climate Center and the Wild Me nonprofit and Wild Road platform that is now part of Conservation X Labs, and many collaborators. And if we do this, right, that's the motivation that when we succeed, the pay will come in the form of improved natural resilience that benefits us all. Thank you very much. Happy to answer questions.
I was trying.
Yeah.
And what made it so engaging? I'm curious to know on how it can deal with the challenge of extinction
Of what?
of invasive species.
Yeah. So it actually, and I'm happy to give a whole lot of talk on the genomics. So actually, cryptic are mostly cryptic to us, not to the camera.
and not to machine learning algorithms. And that's another beauty of using these computational approaches. There are a lot of things that we can deal with computationally that may appear completely indistinguishable to us. I actually have a bet that's going on with somebody who says, oh, you cannot distinguish between all of these rodents. So give me enough data, we will, right? And that's, so we are so biased to the way that we view the world. There's a great paper that I don't have in this talk, but there's a paper that showed in this about a year ago that showed up showing that humans do not have enough red-orange acuity to distinguish between some species of moths, some phenotypes of moths, where machine learning had absolutely no problem. Because we have to remember, right, a lot of it did not evolve for our benefit. It's not our vision, it's the bird's vision that matters when distinguishing moths, for example. We're seeing moths in the first place. We were just at the insectarium and saw that, it's different spectra, different acuity, different ways of sensing the world. There's the umlaut of all the other species is different from ours, and a lot of the species, the traits that we might completely not even be able to see are there because they're perceived by the senses of other species. And so and to me, exciting opportunity that computational tools and like all the other sensors and machine and processing through machine learning allows is that ability. Like, what is it that we're missing? Which may be actually important for this, for the interactions among species and our understanding of what might happen if. And the other aspect, even when we can see like the zebra stripes, if I let me, oops, sorry, right. If I go all the way back to the very first slide that's there in my title, let me get it here. And I ask you, okay, which two zebra stripes are more similar to each other? These two, or these two? No amount of training will let you do it. We're really good at saying, not saying. Okay, like after about 5 minutes, most of the people, we've never even like bothered to look at zebra stripes, will be able to figure out, okay, not the same, or like same or not the same, given two pictures, but to quantify, no way. Right? And yet computational approaches can. So this is not a problem. We can quantify, we can point out where the differences are, how similar. And so that allows us to do, for example, really cool things like correlate pattern similarity to genetics, to understand kin selection and kinship recognition in species that do not use faces, right? Because we're really good at quantifying similarity of faces. Oh, the baby looks like its mom, the baby looks like its dad. But, zebra stripes, I don't know. So yeah, so this was a very long answer that we need to start rethinking of what is it that like really a human limitation versus a true fundamental biological trait, or more broadly, a principle.
Is there a risk with the genomics that we reproduce existing bias towards big and charismatic species when we come to the event like this?
It's a risk if we don't bother changing the status quo. But by the virtue of what came together in the ABC, we're acutely aware of all of these biases, in fact, the biodiversity shortfalls, and Laura led this wonderful paper, watch this spot, hopefully it will be public soon at some point, that really talks through all of these biodiversity shortfalls and how we can address, and in some cases, like David's AMI traps, right, in the whole pipeline of really pushing this kind of the boundary of our insect bias.
So if we waltz in without any understanding of biodiversity and ecology and the space, then yeah, we will. But we're hoping that we're a little bit more, like we have a little bit more humility as computer scientists.
And talk about the bias that the data. I'm curious to see how you think it's sort of that. Like I know there's people working on ways to work with the distribution or like get better data, but in the end, it's about recursion, like you said, like people have recursion take real data, so we don't do research. What's your take on having?
So we are doing new research. So that's a really, really, really hard question, how to deal with this bias. And so the entirety of mission on the computer science side for the AI and Biodiversity Change Global Climate Center, ABC, is really how do we create machine learning methods that focus on the long tail of the distributions, that focus on out of distribution data, right? So the change in what you're actually using your methods for versus what they were trained on, and part of it is, so there's several ways. There's first of all developing methods for dealing with this, and the concepts that we're going for to really be able to deal. Some of this I showed a little bit in, can we use multimodality? Because data are biased in different ways, and can we use multimodality to constrain one modality, to kind of bootstrap, right? To get information out of the modality where we have data, to maybe infer something about where we don't have data. And we need to do it carefully, because it's not an easy task in general, but leveraging the places where we do have data to extract relationships, implicit relationships in data and data modalities that hold and generalize. So for example, why is ChatGPT so good at predicting and at kind of seeming like it's talking to you and like producing something coherent? It learned on a lot of data what are the implicit relationships between the like n-grams of words, right? The co-occurrences of words. That's it. Like fundamentally, that's it. So can we learn from where we do have a lot of data, these kind of implicit relationships between like very fine grained kind of things that we may have not noticed, that may generalize and allow us to impute missing data in places where we might be missing one of the modalities, but do have the other ones. So, there's a lot of, but it's a hard problem honestly, it's a hard problem. And for example, one of the other things that we discussed today is, in many cases, we might not need to be able to predict exactly the species or the population size. Can we predict the change? So there are some things that are actually easier to predict, easier to estimate than, and like we need to figure out what are they, and how do we do that in a way which is believable, if not reliable, right? Yeah.
So in practice, how much models developed by people like OpenAI and Split?
Sorry, how does the big
practice? How much, let's say, the models developed by big companies like OpenAI or Anthropic are useful in the developers?
They're not doing any of it. We're training our own models. They're not using any of this data, right? So that's not what makes money.
Compared to the data models,
yeah, we still need to, because we're adding knowledge, the biological knowledge and structures into these models in a very different way. In some cases, you can start from pre-trained models, but generally, because they're trained on such different data with such different assumptions, they for BioClip, for example, we compared it to Meta's models, llamas, and we compared it to the proper comparison here is CLIP, which is the base, the foundation model, multimodal foundation model, like the other ones, but doesn't have the generative component. It's explicitly trained for multimodal data classification. So, you know, on the tasks, on biologically relevant tasks, these models like CLIP, they bounce spectacularly. Like they, you know, we have a whole paper, happy to geek out on that. But yeah, they don't even provide a good baseline. Let's put it this way.
And we were talking, so you wear different hats. So you're a researcher, also co-founder of a nonprofit, and your talks are really like the example of zebra. So was that done through wildly? If so, or your advice be for a researcher who want to have environmental impact that has another ways than just your academic research?
Yeah, that's a really good question. So if the first thing, don't start a nonprofit. It takes, so not only that, but it's an AI for conservation nonprofit. So it's an expensive nonprofit, like staff, personnel is expensive, resources are expensive. And then, so we need to raise money, and we ran it as a startup. So we essentially, you know, started a startup, which people take time out of their academic careers to do, but then at the end they have millions if they're successful. In this case, we sunk the millions into a nonprofit, which I don't regret at all. And it has impact, and it's like tangible, wonderful, measurable impact, but it's a lot of work. So, and that's really not the only way to do it. The reason we did this is because we were, at the time, 2013, there really were not any, there weren't any AI for conservation, you know, nonprofits, any kind of organization that would take academic research and translate it into tools that conservation community could use. So really at that point, we saw no option but to do something like that. I think at this point, there are plenty of other ways of making impact, of bringing academic research, and ABC, I would think is a good example of it. That really it's use inspired, like impact ready academic research. It's very fun, that foundational academic research, but then there are organizations like Wild Labs, for example, that is a partner in ABC, but it's a tetra conservation community. It's a massive online tetra conservation community that brings together everybody, for example, who's interested in different aspects, everything from how do I configure my camera traps and what's the right drone to fly in this particular region, to does anybody know of the state-of-the-art algorithm that deals with identifying, new species, which is still a very open problem in research. So for practitioners today, like if you want to deliver impact and tools for, for people to use, or scientific knowledge for people to rely on when they make decisions in the field, or when they make policy decisions, there are other ways of doing, whether it's through GeoBond or through Wild Labs, or through now about 100 good leading organizations that are in this space. So partnerships are the place to go, but on the kind of reinforcing that statement, partnerships are the way to go. I think that in this space, doing academic research in the isolation kind of the confines of academic boundaries is not particularly useful. Yeah.
Thank you both. Really interesting, and I'll dig into it a bit more.
But like, if you can make a statement on the state of the research that focuses on helping models understand that this is a novel new species that are not trained on, like mentioned now that it's obviously very difficult and it's active user there's some insight.
Yeah, there are many ways to go about it. So this very active area of research in machine learning in general, it's called the open world problem. And David also can tell you quite a bit more about it. There are different approaches that people take. And okay, and who just left? Hey, sorry, you, yeah, so, you know, if you wanna say a few words about how you approach the problem of the open world. I'll be. Maybe I'll summarize so people can hear, because I don't know how much of it was heard here. The fundamental underlying approach there is taken is anomaly detection or out of distribution. So you really look for things that are anomalies that are in the simplest, most basic way. The anomaly approach is standard deviations away, several standard deviations away from what you would expect. So anomaly detection is one approach. Another approach that we're taking, for example, in Wildbook, where for every species that so far we have in Wildbook, the distribution of the number of images per individual is also this hugely skewed distribution. And so you have lots of images for very few animals and then long tail. And so in fact, more than half of the animals have only one image, and then about 70% have one or two. So majority of the queries, when you have a picture, it's not which one of the zebras it is it a new zebra, right? So it is very much an open world problem. And so we approach it by building a clever data structure there. So there are other ways. So anomaly detection is one. The other one is, can you build proxies in your data that that maybe capture the same not same rather than which one? So those are two very different ways of posing the question. So including species specification, you can ask which one of the species it is versus the much easier problem is if I give you two pictures of organisms, is it the same or not the same at the species or individual level or whatever it is that open set that you have? And that problem, it's much easier one. If you can solve, it allows you to actually then push your ability to do this open world out of set, completely out of set problem, like much better for 90+% of your queries. And then we claim nothing is 100% accurate. So the question is not how can we design methods that can solve the open world problem, but how can we design methods that that trade off what the humans actually need to look at and need to process versus the accuracy of the overall pipeline.